All right, and I think we're live. So welcome back, everybody. We hope you are enjoying the event so far. We have finally arrived at our last chapter, Chapter 3, Health. I think nanotechnology's role in medicine to diagnose, heal, and grow is prevalent and far-reaching. And I'm sure most of you realize that as well. In our polls at the beginning of the day, we looked at the popularity, and clearly many of you are very, very interested in the role of nanotechnology in health. Okay, so let's start with a question. How can we detect cancer at its earliest stages, when it's just a few cells large? Well, first we can look at new types of imaging, or we can send in small beacons that flow through the bloodstream that find their ways to the malignant cell mass. Then, once we've discovered the ailment, perhaps we can send in a tiny machine with nanoscale precision as to not harm the neighboring cells. But how do we make sure these devices can get to their targets successfully? And how do we design such a device? Well, this brings us to the first segment in our health chapter, a keynote talk by Dr. Robert Langer. Unfortunately, Dr. Robert Langer is a busy man, and he could not make it this weekend, but he was kind enough to sit down with me earlier this week to record the lecture and even ask him a few questions that several of you had submitted in the past week. So we tried to maintain that sense of engagement there. So who is Bob Langer? Dr. Robert Langer is one of the 11 institute professors at MIT. His 1,500 articles have been cited over 325,000 times, making his citation impact the highest of any engineer in history. He served as the chairman of the FDA Science Board and has garnered over 220 awards over his career. Dr. Langer has been elected to the National Academies of Medicine, Engineering, Sciences, and Inventors. We are very pleased to present you Dr. Langer's keynote address.
Thank you so much. It's a pleasure to be able to speak to you today. What I'd like to do is start out about how I got started in this area, and then tell you about some of the more recent things we're doing. So if I could have the first slide, please. When I was I'm a chemical engineer, and when I finished my degree, I did something that was very unusual for a chemical engineer. Almost all my friends in chemical engineering went to oil companies, and I wasn't that excited about that. So I ended up going to Boston Children's Hospital and working with a man named Judah Folkman, who had an idea that's shown on this slide. And interestingly, I was the only engineer in the entire hospital at that time. This was 1974. Now, his idea, which is shown here, is how tumors grow. And basically what he said is that somehow a tumor cell would become abnormal, and then it would grow to a three-dimensional mass to reach maybe about 1 million cells. And this is depicted in the bottom middle of this slide. And when it gets to that size, say 1 million cells, it runs into a nutrition problem. The cells in the center die because they can't get nutrients or get rid of waste. And so they wouldn't get bigger than 1 millimeter cubed or 1 million cells. But what the tumor does to solve that problem is it sends out a chemical signal, which Dr. Folkman called TAF, or tumor angiogenesis factor. And what that does is it causes surrounding blood vessels, which normally don't do anything, to start growing directly to the tumor. And that then solves the nutrition problem because the nutrients can get in through the vessels. And it also enables the tumor to succeed. And as you see in the upper right, once the vessels are there, that now the tumor gets bigger and bigger and bigger. And if you go to the top, you see that the tumors could actually spread through those blood vessels. That's a process called metastasis. That's often what kills. And Dr. Folkman had this idea that if you could stop the blood vessels, achieve what's called anti-angiogenesis, maybe that would be a whole new way to think about trying to stop cancer. Well, this was apparently a very speculative idea. Since I didn't know anything about biology, I thought it was wonderful. So, and Dr. Folkman asked me, my job would be to actually prove he was right, but also in so doing to isolate the very first substances that could stop blood vessels from growing. So I would isolate different substances from cartilage. But the key limitation to all this, and this is true for almost anything in the medical field when you're finding a new factor, is having what's called a bioassay. And there was no bioassay for studying blood vessel growth. So we had to invent one. And what we did is we thought about using the eye of a rabbit. And And so what we did is we put certain types of tumors in the rabbit's eye, what are called V2 carcinomas. And normally, of course, there's no blood vessels in the cornea, that part of the eye, either. But when you put the tumor in, as I'll show you, blood vessels will grow. But it takes maybe 9 weeks for that to happen. So we need to have a little drug delivery vehicle, like a micro or nanoparticle, that we could take whatever it is that we isolated and slowly deliver it for this 9-week period. And so we ended up asking different people if they could help us. And we even looked at the, and everybody said it would be impossible. The literature also said it would be impossible. And this is an example of which is shown in the next slide. And so basically people thought that you could only get small molecules to get through these materials. And maybe the only thing that we had going for us is that, we hadn't really read the literature. But the idea that people were concerned about is that if you had a big molecule, how could it pass through a solid object, so to speak? Well, at any rate, I spent maybe 2 years working in the laboratory, experimenting kind of what I'll call Edisonian-like with different approaches, different materials, different ways of making them. And I actually found over 200 different ways to get this to not work. But eventually, I was able to make little microparticles or nanoparticles even. Next slide. And you see a picture of one of these on the top left and also on the bottom right. And with this one approach, I was able to show that you could actually release almost anything that was a macromolecule for as long as you wanted. And the next slide shows a graph from a picture, from a paper rather, that we published in the journal Nature in 1976. Now the release rates weren't constant there, but later on we figured out ways to make them constant. And in that same paper, we not only showed that you could release molecules like proteins or peptides, but it was also the first demonstration showing that you could encapsulate things like nucleic acids, like DNA or RNA, into these tiny particles, something I'll come back to later. Nonetheless, when I first presented this work, it was met with enormous amount of skepticism. I remember in 1976 presenting this to a big chemistry and engineering audience, and I was very nervous. And when I got done, I stepped off the podium, and I felt actually okay because I didn't forget too much what I was going to say. So I thought when I was done, all these older scientists being nice people, they would encourage me, this young guy. But when I got done, I stepped, as I said, I stepped down, and a whole bunch of them came up to me and they said, "We don't believe anything you just said." In fact, the next slide shows what they said. They said that these large molecules can't slowly diffuse through solid polymers, and organic solvents, they'll destroy whatever you put in. So that was very discouraging. And actually things went downhill from there. I tried to get a job in a chemical, I tried to get an assistant professorship in chemical engineering department, and no chemical engineering department would hire me. They basically said, this bio stuff you're doing doesn't make any sense. And then I tried to get grants, and my first 9 grants were rejected. The reviewers would say, well, Dr. Langer doesn't know anything about biology or cancer. And so finally, I got a job in a nutrition department. And the reason I got it, I think, was because the guy who was in charge of it, he was like what I'll call almost a benevolent dictator kind of person. He liked me, so he offered me a job, but he didn't bother to ask the rest of the department what they thought. Still probably would have been okay, except for one thing, which was that which was that the year after I joined, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. In fact, the next slide is from a talk that my friend Michael Marletta, who later became chairman of Berkeley's chemistry department, a member of the National Academy of Science. Mike was giving this talk actually about me a few years ago, and he was referring back to 1980. And he said one evening he went to a faculty dinner at a Chinese restaurant with me and some senior professors. He said a senior scientist sat quizzing us while smoking a cigar. And he said, when the older scientist heard my concepts for polymeric drug delivery, he blew a cloud of smoke in my face and said, "You better start looking for another job." And Mike said he thought he was in a Fellini movie. So that wasn't going so well. But, I kept at it, and and eventually, as I mentioned, we were able to show that you could use these technologies for drug delivery. And so we were able to use this also to try to solve the angiogenesis problem. And just to go back over what the assay was that we were trying to develop, that's shown again on the next slide. And you see the tumor and the polymer. Now, I want to not show the next slide right away. I want to give a warning. If anybody is squeamish and doesn't like the sight of blood, you shouldn't look. But what we did, and this was enormous amount of work with some of the surgeons, is we basically, took 200 rabbit eyes, 100 fractions, and tested them. And every time we did a fraction that was inactive, as I'll show you, the blood vessels made these sheets, the tumors became three-dimensional. But we did find one fraction that stopped the blood vessels. And this was really historic in a way, because that had never happened before whenever anybody did these kinds of tests. And the next slide now will show representative ones, not even our best or worst, from a paper we wrote in Science in 1976. And what you see on the upper left is 9 weeks after the start of the experiment, you see a sheet of blood vessels growing over the polymer on the way to the tumor. If you looked at this eye 3 weeks later, it would be three-dimensional, out of the orbit of the eye. It would actually kill. But when you put the inhibitor in, and we did this 21 times, and it happened all 21, the blood vessels are, now you look in the lower right, the blood vessels are lower, they're sparser, and they totally avoid the polymer, which was striking. And actually 60% of the time, these tumors never grew. So this was the first time showing that you could actually isolate angiogenesis inhibitors and that they really would stop blood vessels from growing. Nonetheless, it took another 28 years from this paper before the first angiogenesis inhibitor was approved by FDA. And these are now shown on the next slide. This was quite striking. Like I said, this paper is 76. Now we're going to go to 2004. And even though it was a long wait, what happened was amazing. One after the next blood vessel inhibitors approved by the FDA. And Avastin, the first of these, also the last of these, is actually the number 2 or 3 largest selling biotech drug in history. And there are many, many others that have been approved for other kinds of cancer. And not only have they been approved for cancer, but they're very bad eye diseases. Some of the most common eye diseases in the world: macular degeneration, which causes blindness; diabetic retinopathy, which causes blindness. And now, and before this, there was never a drug that existed that could stop those diseases. This, for the first time, drugs like Eylea and Lucentis are now widely used. They're also blockbuster drugs, which can, in some cases, even reverse these. So, so this was very exciting to see this happen. Now I want to go back to the 70s, and Dr. Folkman and I talked about this, and we said, gee, this is exciting. We can deliver these molecules. Maybe that could be useful for various drug delivery applications. So we said, well, maybe we should file a patent. That might seem common today, but in the 1970s, Boston Children's Hospital, where I started working, never had a patent. So we filed for a patent the first time. And 5 years in a row, the patent examiner rejected it, and which was very discouraging. And the head of the technology transfer office there at the hospital came to see me one day and said, Bob, you're wasting an enormous amount of money. This guy is never going to allow this patent. You should just give up. But I don't like to give up. So I started to think, is there some way that I could convince the patent examiner, legally, of course, to allow the patent? And as I mentioned in the beginning, when we first started doing this, everybody said it was impossible. It could never work. So I wondered, did anybody ever write that down? So what I did is now 1982, this is after 5 rejections and 6 years after the Nature paper, I did what's called a science citation search. I looked back at our 1976 paper to see who wrote stuff about it. And I found a number of papers, but I want to highlight one on the next slide. And this was from 5 of the top chemists in the world. And what they had said, they were reviewing this field in 1979. I had no idea this had been written until I did the search. But they're reviewing this, and they said, well, generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates through polymers. However, Folkman and myself have reported some surprising results that clearly demonstrate the opposite. So I showed this to the head of the tech transfer office, and he was very excited. He flew down to Washington to show it to the examiner, and the examiner said, gee, he said, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from these 5 people that they really wrote this. So I wrote each of them, and they wrote me back. They were nice enough to write me back that they really did write it. And I showed that to the head of the tech transfer office. He flew down to Washington to show it to the examiner. And basically we got this patent shown on the next slide. So it's a very broad patent, and we use that patent to teach different people how to do this. I got involved in starting companies. We licensed it to companies. And the next slide shows that there are many, many products based on this that are now used all over the world by tens of, if not hundreds of millions of people. New microspheres that can deliver peptides to treat prostate cancer, endometriosis, new peptides to treat type 2 diabetes, ways of delivering schizophrenic drugs, ways to combat opioid addiction, drug-eluting stents, all do new ways of treating eye diseases, brain cancer, and so forth. So that was very exciting. All of these systems, by the way, they're injected underneath the skin, say subcutaneously or intramuscularly. But we also thought that we could adapt these approaches to inject right into the body, to solve different problems and maybe make what I'll call targeted therapies. Now we'd have to do it by nanoparticles. But the next slide shows what I was thinking about, which is that you could certainly change the payload. People had made single molecules for targeted therapies like Humira. But if you were able to put if you were able to put a drug in a nanoparticle, your payload is much greater. In a single nanoparticle, you might be able to put anywhere from 1,000 to 100,000 molecules in the nanoparticle. So if I picked a hockey analogy, so what we did is we wrote on the blackboard, next slide, all the characteristics we'd want in such a nanoparticle. High encapsulation amount, high encapsulation efficiency. The difference is in the first case, you want a lot in the nanoparticle. In the second, you just don't want to lose a lot when you're doing the encapsulation. Ideally, it'd be great if they could be freeze-dried and that they don't aggregate, that they do degrade, that they're small so that they could be taken up by a cell, and that they're not rapidly cleared by, say, the liver or macrophages. So what we did, next slide, this was led by one of my postdocs, Ruxandra Graf. We picked polyethylene glycol, which is a coating that's kind of attracts a lot of water. So our thinking was that maybe it's going to be a disguise because the cells like the macrophages, they see water anyhow. Maybe they're not going to eat it or gobble it up. So we made these. We published the details in Science. The next slide just shows that by quasi-elastic light scattering that they are in the nano range. But what the next slide shows, which was very exciting, is that if you incubate these with macrophages without PEG, and you put an orange dye in them, on the upper left, what you see is an hour later, there's all these little particles in the macrophage. But when you put the PEG on it, and we did different variations, like a single chain with different molecular weights, or even a triple chain where we got perfect coverage, there was either no or very little uptake of these polymeric particles. So then what you could also do, and this was now done by Omid Farakadazad on the next slide, is actually put a targeting molecule on these. Now, I realize that, I'm explaining this, and I don't know that I'm explaining it very well, but I was lucky a number of years ago that Nova, a TV show in the United States, one day came to film some of what we were doing. And they actually made a video of what we did. And they explained it so much better than I that I thought I'd show you that video instead of me explaining it. So if we could put on the next slide.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
So basically, I should point out that a lot of clinicians that we work with tell me it doesn't blow the cell up quite that way. But the beauty of this is that you could put small molecules in, which we looked at for cancer treatments, and I'll mention that in a second. But you could also, and we've done this as well, because of what I mentioned that we started even back in the 1970s doing, you could put, with variations, things like siRNA or messenger RNA in. And many groups, some in Canada, some in the US, have greatly advanced what we did on nanoparticles. But the beauty is that you could put all these kinds of molecules in. So, for example, I helped Alnylam company I'm on the scientific advisory board of. We put siRNA in these nanoparticles, and these were actually lipid nanoparticles, and we got approved by the FDA for treating a rare disease called ATTR amyloidosis. And I started a company called Moderna, helped start a company called Moderna, and they put messenger RNA in. And one of the messenger RNAs is the basis of the COVID vaccine. So all these end up being in different nanoparticles. Now, if we could go to the next slide. So for small molecules, we're seeing different anti-cancer drugs being used in some of the different polymer systems by AstraZeneca, Pfizer, Selecta. But one of the things that Dan Anderson, who was one of my fellows, now a professor at MIT, and I did, so we said, there's not that many lipids that people are using. Maybe we should increase the number of lipids. So we made this idea that maybe we could make a library of lipids. So at any rate, what we put on, again, is another wish list on the next slide. And so our wish list was that we could use combinatorial chemistry to make literally thousands of lipids. And the wish list is one protocol to do this, make a very diverse library, as I'll show you, in very few steps. You don't want to have protection or deprotection or catalysts or solvent switches, no purification, and so forth. So let me just show you some of the chemistry that we did. The next slide shows a picture of the R groups that we use. So tremendous chemical diversity. And the next slide shows that what we could do is change the number of tails on the left. The length of the tails on the left, or even the number of tails on the right. And like from 5 to 4 to 3. And we used different chemistries, basically like a Michael addition, to actually make literally thousands of these different lipids by combining, different diamines and so forth. And basically, what we did when we did this, we had this library of literally 1,000 lipids. And we worked closely with Alnylam, where one of my students, Kenna King, worked. Now he's vice president. And let me just show you part of what we learned. The next slide shows a dose response curve, which is good. I'm sorry, not just a dose response curve, but it shows you what happens over 30 days, that you keep on getting a factor 7 made. And the next slide shows that this is a dose response curve, whereas we kept increasing the amounts of factor 7, you get get knockdown. That's what you want with siRNA. And when you go to 5 or 10 milligrams, it's almost gone. And then what we did is we took our best ones from this library and then used an epoxide chemistry to make the best ones hopefully work even better. But before I show you that, I want to just show you how what we did is you could not only knock down factor 7, which is like a marker, but you could even knock down lipoproteins that might give you hypercholesterolemia. And that's shown on the next slide. And here you see on the right part that you can get actually almost a 50% knockdown of LDL. So let's say you're at a very high cholesterol level, this might be a very good thing to do. Now, as I mentioned, and you can keep doing it over and over again. It's very reversible. If we go to the next slide, you can see that you can do it like once a month. And as I mentioned, we also wanted to make these better, make them more potent. And what we did on that is shown in the next slide, that we could use take epoxide chemistry and select amines from the original library and now make even a better library. And some of what we found is shown on the next slide, which was very exciting. So the one we did earlier, we called LNP-PO1. And you can see that's in the mg per kg, 1.5 to 6 mg per kg on the right. But if you use this new one, which we call C12-200, notice how you can go down even with 0.003 mg per kg, you still get significant knockdown. So this is about 2 orders of magnitude better. And what that means is that you could actually, treat a disease like ATTR amyloidosis. And here on the next slide, you see some data in primates showing again that you get very, very significant knockdown, even with as low as 0.03 mg per kg. And we worked closely with Alnylam for many years, really trying to make these nanoparticles more and more potent in terms of giving knockdown. And the next slide shows what we did collaboratively with Alnylam. So when we first started, you want to look at the right, and it shows you what you needed to get a factor 7 knockdown. So in the beginning, we needed between 1 and 10 mg per kg. And then we just kept improving it, either us or them, using these combinatorial approaches. But if you go all the way to 2014, and now you look at Izzo Dong's paper, what you can see is you probably get about a 4 order of magnitude improvement, which is so you, and that has a huge effect on improving toxicity. By the way, Alnylam, not just because of this, but they now have 3 products approved by the FDA using siRNA. So a whole other thing that once we tried to do is on nanotechnology, and this was totally serendipitous, was done by Armin Shari, one of my PhD students who worked with me and Klaus Jensen. And we were doing, Klaus and I wrote a grant to the NIH, where what we were doing was seeing if we could understand better how molecules could get into cells, particularly genes. And if we go to the next slide, what you see is just a cartoon. And some of the ways that people tried to do it are electricity, cell-penetrating peptides, nanoparticles, liposomes. But if we could press the button for the next slide, so to speak, what you see is each has disadvantages, like damage or toxicity. If we could go to the next slide. So what Armin did, if we could go to this video and turn the video on. So Klaus was an expert at microfluidics. So what Armin did is we made a microfluidic chamber with a tight constriction, and we passed cells through it. And on top of that little thing, you see a gene gun. And basically what we do is we keep putting the cell through that constriction, and when it was through the constriction, we'd hit it with the gene gun and just tried to study it. But one day, Armin had an interesting idea. He decided to take away the gene gun. And interestingly, he got exactly the same result. In other words, you didn't even need the gene gun. Somehow, by squeezing the cell, you create nanopores or something like that. And when you do, molecules can get through. So that was amazing for us to see. We published this in PNAS. And then next slide. So what Armin then did is he built some of these devices. We actually even started a company on this called Squeeze, not surprisingly based. And when we initially did it, you could get 1 million cells per second. Now we've got 100 people at the company. He's actually the CEO. You can actually put 10 billion cells per second. It's really quite remarkable. And it actually works, as far as we can see, quite well. The next slide just shows an example of one of the studies he did. He actually compared efficacy of conventional ways of inserting things in cells, like cell-penetrating peptides and nucleofaction, which is the same as electroporation. And basically what he did in this case was take what are called the Yamanaka factors. These are genes that can convert, like to say, a fibroblast to like a stem-like cell. And the way to judge that is by colonies. And what happens is you get 2 with the cell-penetrating peptide, 11 with electroporation or nucleofaction, but 150 with his device. And interestingly, Scientific American, next slide, in 2014, named their top 10 world-changing ideas of 2014. And they picked CRISPR-Cas9 as the first one and squeeze as number 2. I felt they had the order wrong, but it's okay. And not only did this end up being more effective, it also is safer. You get less cell death. And what was really striking, and this was a recent paper Armin and his team published, next slide, is if you look at what's called the transcriptome, that's the messenger RNA profile, and you compare it, it's like a gel, to the control, to electroporation, which is the gold standard, to the squeeze approach. And the electroporation was even mild. You get much bigger changes with electroporation than the squeeze approach, which I think will have someday maybe significant regulatory implications. So I thought I'd end the talk by telling you about one other idea where we made little microchips with nanowells. And this was done by John Santini, one of my graduate students at the time, along with Michael Sema, who's one of my close collaborators. And let me just show you a picture of what John made on the next slide. So this is a microchip. It's all done by microfabrication. We publish this in Nature. You can actually have these wells where you put active substances in and then cover them, say, with gold or platinum alloys. And they'll stay in there. They'll be stored in there and in the well until you take the cap off. And you can do that by an electrical signal. So let me just show you some pictures. Next slide. So here we have a picture of one of these early chips that John designed. You see the top view on the upper left and the bottom view on the right, upper right. And next to it's just the United States dime, which is our smallest dime. Now this is a short flat chip, but as I'll show you soon, you can make them any size and shape. You can even make them injectable. Now the way they work, next slide, let's just look at a particular well. You see a scanning electron micrograph of the well. It'll stay like this, even in the body, for a long time. But you could come along and just apply 1 volt externally. And what will happen in nanoseconds is the cover will come off. Next slide. And you see this starts to come off on the left and is fully off in the right. And that's way less than a second. And then once that happens, the drug can come out. And just to show you a couple of graphs from that Nature paper, the next slide shows if you put different amounts, you want to get different amounts out at different times, you can do that for a single molecule, but you could even envision this sometime as like a pharmacy on a chip and deliver multiple drugs. And that example of that in vitro is shown in the next slide, where we put 2 in and made them come out at different times. And we've actually taken this to the point where we put it in human patients. The next slide shows a clinical trial that we did in Denmark. And you can put the chips in patients and you can communicate with them by what's called the Medical Implant Communication Service Band. And it's approved by, in the US, the FCC and the FDA. Now, you can actually even have a special computer code if the patient or doctor wants to change the dose. And these are very well encrypted. We actually had somebody who had worked on the air-to-ground missile working on this, and she said this was better encrypted than that. And not only do you have that, but you have a bidirectional communications link between the chip and the receiver. The receiver, by the way, could just be like a cell phone, and it tells you everything. Did you deliver the drug? How long will the battery last? And so forth. Well, in the first clinical trial, what we did, next slide, is we did 8 patients, and we wanted to pick a disease that we felt maybe someday could make an impact. And the disease we picked was osteoporosis. And right now, one of the main ways of treating that, and this is often a disease for elderly women, is you give injections of what are called parathyroid hormone, a large peptide, and you give it once a day. You actually don't want to give it continuously, because if you give it steadily, you get bone resorption. So you want to give it in a pulsatile fashion. Problem is, if you inject it once a day, less than 25% of the people stay with that. So it doesn't really work. So what was done in this trial is a small office procedure to do the implant. And what was interesting is we got the same pharmacokinetics with actually less variability. And then the three types of measures, calcium and PNP and CTX of how you're treating it, were the same as daily injections. And just to show you some data, next slide, is here you're looking at human data at day 60, 68, 76, and 84. And notice the graphs, they just superimpose. This is human beings. And just to show you again some pictures, if you take the device out, you do get fibrous encapsulation, as you see in A and B in the bottom right. And that's a big device. We've now made them much smaller. But if you look at histology on the left, you don't get any immune cell. You don't get any cells like macrophages. You just get a small capsule. And as you can see from the graph, that has no effect on what comes out. And now we're working actually with the Gates Foundation and using these for a new method of birth control for helping poor families do family planning and things like that. And a whole other idea we thought is that maybe we could use a variation of this for what's called precision medicine or personalized medicine for treating cancer. And just as an example, you know, I'm sure everybody realizes that everybody's cancers are different. So one of the ideas that's been put forth is that maybe you could do a biopsy of a patient's cells, take them out of the body, and then grow them in vitro and hit them with maybe 100 different drugs and see what works the best, and then go back and treat the patient that way. And that's, I think, an exciting idea, but it misses certain things, right? It misses the immune system, which is very important, misses blood vessels, misses the extracellular matrix. So we thought maybe rather than take the cells and bring them to the lab, which is what everybody's been doing, maybe we could do the opposite and bring the lab to the patient. Well, how could you do that? Well, let me go to the next slide, which is a variation of what I just showed you. What if we made this chip more like a cylinder, a very, very tiny cylinder, where we have micro or nano wells? And in this, let's say what you do is you could actually go in and take a biopsy needle, like a 19-gauge needle, and fit this little now chip, which is a cylindrical, right in it. And let's say you make 40 or 100 wells in it. And each of those wells, you put 100 different drugs or 40 different drugs. So the idea is you deliver it, to the site by biopsy needle, and you leave it behind. And then in the next 24 hours, it delivers, where you see dosing, it delivers microdoses to the tumor. We probably put 2 or 3 of these or more in the site. Then the third thing is retrieval, which means 24 hours after you started the experiment, now we come back with a slightly bigger needle, and we take out not only our device, but a little bit of the surrounding tissue. And then on the final panel, we get a readout, and we look at apoptosis. How did the drugs do? Did they kill the tumor cells? On the next slide, what we're showing is just a picture of this tiny device next to a small coin. And the idea is you could test 30 or more drugs in parallel. You get an answer in a day or two. It's minimally invasive, and you're doing that anyhow with biopsies. And you're not getting systemic exposure to any drugs. And you're only giving 1/1,000,000th of the systemic dose. And the next slide just shows a picture of how you can watch the drug come out over time by fluorescence or multi-mass spec. And then Ollie Jonas, he's the one that led this project. We tested this in a number of models. There's been about 15 publications. And I'll just show you one in the next slide, which is published in Science Translational Medicine. So the question is, if you take TNBC, that's triple negative breast cancer tumor model, and you hit it with 6 different things, just 5 drugs and a control, what apoptosis do you get if you give them systemically? And how does that compare with the device where you're actually giving it locally with 1/1000th the dose? And you can see that they're pretty much identical, same trends. So this starts to tell us that you might really be able to use an approach like this to develop personalized cancer treatments along these lines. And Ollie now is a professor at Harvard Med and is doing clinical trials with a variety of the big cancer hospitals like MD Anderson and the Farber and Sloan Kettering. And again, it's very early, but we hope that this could be a very exciting way of using this kind of microchip nano approach to develop personalized treatments. So I want to thank you so much for listening to me. It's been a pleasure to speak to you, and I want to wish you the best of luck at this conference. Thanks so much.
Yeah, so over the past week, we received about 40 questions, and most of them fit into a few categories. But to summarize, the first question I have is, where did it all start? So you started your presentation off with your work in Dr. Folkman's lab, but was that the first time you decided that you would be journeying into the field of biotechnology, or did you have some inkling of interest before then? Maybe in undergraduate or even in high school? And how did you know where to go next after that?
Yeah, well, I always liked the medical things, but I like chemistry, and that's why I went into chemical engineering. But I did my PhD in the bio area, and then and Dr. Folkman, that was, I mean, it just changed my life. That was just such an inspiring experience, and he was a wonderful mentor. And I thought the angiogenesis, area was just very, very exciting.
Right. Thank you. Several questions were asking about the accessibility of nanotechnology. So do you believe that nanotechnology will be accessible to everyone in the future, just as now everyone has access and local control on the internet? Like when people hear the word nanotechnology, thoughts about very complex math and physics come to mind. So do you think there is a way to abstract this complexity so that everyone everywhere will interact daily with nanotechnology?
Well, I think yes and no. I think nanotechnology, you may interact with it. Like I said, the COVID vaccine, all the COVID vaccines are probably going to be, or at least the mRNA ones, and also the Novavax ones are nanotechnology. So a lot of people will get it, but I don't know that people even realize that. It's like everybody keeps talking about the RNA part, which is obviously very important, but and is key. But nanotechnology manifests itself in all kinds of ways, and you wouldn't be having the mRNA vaccines if you didn't have nanoparticles.
Right. That definitely makes sense. Great. So next, looking forward, you mentioned Scientific American's top 10 world-changing ideas in 2014. In 10 years, what new technologies will change the way we diagnose and heal our bodies? Said another way, in your lifetime, what is one technology that you want to see come to fruition?
Well, I'd love to see all kinds of things. I mean, I think if we had better imaging, for blood vessels and cancer, that would be fantastic. But I always think the answer to that question is it's something that hasn't been discovered yet. In other words, it's something that have a name. If you looked at CRISPR if you asked somebody about CRISPR 10 years ago, they wouldn't have known what it was. Now everybody talks about it. And I think that's good. I mean, some of these things have a label, but then people come along and they make a discovery or a finding, and that becomes important. And so I think that to me makes the most sense.
I see. And so what steps do you think our researchers and academics will need to take to discover that next thing? So for current undergraduates or graduate students, how do you get those creative inspirations for pushing the boundaries?
Yeah, well, I really think it comes through basic research, curiosity-driven research, and it doesn't necessarily come, as you could see even with my own research, it didn't, all, like in a way, the microparticles that we developed and variations of them and improvements of them are now being used for COVID vaccine. Well, COVID, nobody knew in 1974 that any of that would happen. But, the research doesn't follow a preordained path. So I just think it's really good to think about what's really exciting and important and interesting and drives your curiosity, and then you go for it. And think about the field could have broad implications.
Right. Yeah, that's interesting. So do you think that your background, which was not necessarily in biomedical engineering during your undergrad, you came from a chemical engineering background. Do you think having that extra exposure to a different field helps you be more creative in biotechnology research?
I don't know. I think it helped in some ways. It may have hurt in other ways, but I'm glad I did it. It's certainly given
Right.
me different probably than a regular biologist or a regular chemical engineer. But I don't know. I mean, people have done great all kinds of ways, so it's hard to know.
That's fair. And I think we can finish it up with this last question. So if you had to pick one thing that you would like people to come away from this keynote, what would it be?
Well, I've ended up giving a lot of commencement speeches, and the way I use my career as an example of that, in a way. And my message in that, and I'd say my message here, is regardless of any science that I talked about, I think what's important for people to do is to dream big dreams, dreams that can change the world and make it a better place. But recognize that if you do this, the road may not be easy, and you may get a lot of objections and criticism. And that gets to my second part, which is don't give up. I mean, don't give up on those dreams.
Right. Yeah. And I think your journey is a true testament to that. You spoke about the many challenges and resistance barriers that you came across in your years in academic research and trying to prove angiogenesis inhibitors. So we're all very grateful that you broke through those barriers and really changed the world in incredible ways. So,
well, thank you very much. It's been an honor and a pleasure to speak to you. Thank you.
All right. Thank you, Dr. Langer. Your story today has taught us not only the potential of nanotechnology in medicine, but also what it means to persist through the toughest challenges. Dr. Langer's work 50 years ago is now being applied to one of the most promising COVID-19 vaccine technologies. I think his story is a great lesson for students and current academics, that your research could have great implications for different fields that may not even exist. So with that, I would like to introduce our next segment. So this aims to tackle a slightly different challenge in medicine. How do we regenerate functions to our body that we lost? Our regenerative medicine panel will explore this question over the next 30 minutes. I would like to welcome to the stage Dr. Lee Katamusini, Dr. Mir Sheikhi, Dr. Michael Sefton, and Dr. Samuel Stoop. So whenever they are ready, we will see them soon.